that was to this day, except for maybe 18 days, just about 40 years ago. So that was a long time ago. And back when I joined Herb's lab in the early 2000s, one of the words he would oftentimes use to describe his own science was the word esoteric. And as usual, as a student of Herb's, he used words that I at least needed to go look up at the end of the day. And for those of you like me who didn't know what that word meant, it means that it's intended for or likely to be understood by only a small number of people with a specialized interest. And so that's how he described his own science. And I think, to me, that really speaks to one of Herb's principles, that now, having known him for quite some years, I think to Herb, one of the most important things that drives his science is a search for truth. Whether it's gonna make him famous or not matters not, but searching for truth is really what I think he is on a quest for. But whether it's esoteric or not, 40 years later, this paper really spearheaded a field that, as I said, has ballooned and is now a very, very active field of research, using the Mussel's molecular tricks to try to develop both new underwater and soft adhesives, as well as energy dissipative materials. And Herb has obviously worked with a bunch of famous people along the way. Him and Jacob Israels really had some beautiful papers together studying the fundamentals of this. And today, Herb will tell us about even more of the recent discoveries that he has made in uncovering some of the truths of the Mussel. So without further ado, I'll give it over to you, Herb, and we're very, very happy that you can share new truths with us today. So thank you.
Thanks, Niels. Very amusing and gratifying introduction. And it saves me some words because I will continue that story.
Herb, before you get started, sorry, I forgot to say, of course, if anyone has questions urgently throughout, you can drop it in the chat and I'll pay attention to it. But if it's not urgent, let's wait until the end. And when we get to the end, I'll monitor the chat if you want to ask questions in the chat, or you can raise your hand and I'll call you out. Okay, thank you.
Hello, everyone. I am going to speak about phase-dependent redox properties in Mussel adhesive proteins. That's probably more generally applicable to other adhesive systems from nature. The work that I'll focus on is the product of two very diligent and industrious hands that have finished up and moved on to industry. Eric Valois at Asylum and Razi Mirshafian at Zymergen. I'll also be able to take care of the credits at the beginning, that much of this research, I would say more than 3/4 of it, was supported and sponsored by our MRSEC that is still ongoing, though I'm no longer leader of it. That's shifted to Matt Helgeson and Megan Valentine. Those of you who know me and or not know that one of my model systems of the 5 or 6 that I've worked on in my lifetime is the Mussel and how it attaches. This is a dynamic process that involves the production engineering of many threads, hundreds sometimes, that are radially distributed. And they're referred to as byssus, which is just a Greek word that was adopted incorrectly, it turns out, from an early translation. And it means a holdfast. This holdfast is particularly interesting because it involves extracellular matrix material that's been displaced outside the confines of living tissue. So the Mussel can engineer the direction of each thread, the length of each thread, and even the tension on it. These are collagenous threads, and that's a close parallel to extracellular matrix. However, the distal adhesive ends are largely non-collagenous, and they contain the proteins that I'll focus on. And there's a zoom of a cluster of those here. James Weaver would say not very good production, but I have a better shot later on. So this mussel is the abundant one in California, Middleus californianus. And it's from another engineering perspective, it's interesting because this is a prosthetic material made by the mussel for itself. It's polymers, no living cells, but it does have an insertion point on the inside that involves an interface between this polymeric stuff and living tissue. And that interface has also interested me for a number of years. So the research is very extended. It involves a lot of biochemistry, some mechanics, and some attempts to translate. But in my old age, I'm increasingly feeling like this popular classical composer named Giacomo Rossini, who tried to compensate for his omissions and etc. With some pieces he wrote in his old age and referred to as sins of my old age. I won't call my omissions sins. They're more gaps than sins. And the gaps are caused by two things that are part of the process of doing research nowadays. The constant emphasis on low-hanging fruit and funding agency translational bias. Funding agencies, unfortunately, are not interested in fundamental science unless you can really jazz it up for them. And it's the combination of these two that's led to gaps in my research that I want to do something about. Gap number one is the reconciliation of these two boxes. Box number one shows a surface forces apparatus characterization of the adhesion energy and force of the premier interfacial adhesive protein from the mussels. I'll tell you later what that is, but you don't need to know that right now. And there are approach and separation cycles for several runs here that seem to plateau with this black curve, and the force and the energy corresponding to this can be calculated to give an adhesion energy of 15 millijoules per meter squared. So what you do is you coat one of these microsurfaces with a protein, approximately a monolayer, but that's hard to engineer given that this is an intrinsically disordered protein. And you bring it down onto the other surface in the approach, and then you measure the resistance to separation. If you haven't dealt with these sorts of numbers before, you might say, so what? Well, it turns out that the SFA measurement of the force between biotin and avidin is 10 millijoules per meter squared, and this is above that. So this is really quite a performance at a molecular scale level. So that's one box. The other box just involves allowing a mussel to attach its plaque to a smooth glass slide, and then using a very elegant rig that's described in this article in Soft Matter to pull on a short tether attached to the distal thread of this plaque that is notched at one end. And you get a force distance curve that looks like this, and the fracture energy conservatively speaking, is 100 joules per meter squared. I say conservatively because this is a very unusual morphology for doing a fracture energy test. So I want to emphasize that I recognize that these two energies are not measurements of the same thing, but the fact that they are so different is astonishing. The GC is 7,000 times greater than the energy of adhesion of MFP5, okay? And obviously having more substance here contributes a lot to energy dissipation in this system that just isn't available in that system. So that's gap number one. I need to reconcile those two streams of data. Gap number two is based on experiments done by Jing Yu in 2011, in which he applied an adhesive protein again to one mica surface, brought it down to the other mica surface, and measured a max. However, he changed conditions with each run, going from low pH to approximately seawater pH. And you can appreciate that the energy and the force of adhesion fall off rapidly as you increase the pH to almost nothing at pH 7. Well, we know that mussels adhere strongly at pH 7.5. So this reductionistic experiment has led us to a paradoxical outcome in which the protein has no performance at pH where it's expected to perform. pH 3 is relevant here because the proteins are deposited at acidic pH, but no one expected this loss of performance, and an explanation of that loss is absolutely critical to any kind of translational engineering. So the neglected aspects that led to those gaps were a failure to appreciate the contribution of architecture and a failure to appreciate important phase transitions in the processing of this adhesive. Plaques have a lot of architecture. Here's a mussel with a radially distributed set of byssal threads. Here is a zoom by James Weaver of a plaque showing an elliptical shape. The outline can vary from circular to elliptical. They're in a hurry when they do this, so shortcuts will be taken. And if we cut a section through this and view it with scanning electron microscopy, you can see a protective cuticle that's been cut away. You can see tensile elements that are coming down from the thread and are rooted in this porous matrix like roots of a tree. And there is the porous matrix then that we need to zoom in on, because it has porosity, but the porosity has two distinct length scales, a 1 to 3 micron length scale that involves open cells with channels to other pores, and a smaller 100 to 200 nanometer length scale that is a little too hard to see at high resolution, but it's there. When one of these is made by the mussel, it uses its foot as a dispenser of everything. And the foot is not only extremely compliant and extensible to make all those different lengths of thread that you will observe in a natural ensemble, but it's also like a die cast system in that it has a groove and a depression that it closes off during the formation of a new thread, and the components are pumped into that groove, or actually more accurately drawn into it by vacuum, and they're molded there until they reach a certain stage of maturation, and then the foot groove splits open and a new thread and plaque emerge. So this is a new thread and plaque, and this is another one that preceded that one, until the muscle has hundreds of them. And when does it stop? Muscles have a very strong sense of paranoia that is counteracted by the number of threads that it has. So there's an interesting interplay there. We can get muscles to make threads now for treatment, individual threads for treatment. And these threads would be collected within 1 hour of their manufacture, and then they'd be submerged in buffers of different pH strength. And you can see that the porosity is largely missing, though there are some dimples that are apparent in the pH 3 plaque. At pH 6, you have porosity, but the average larger pores are smaller than they are at pH 7. So there is a phase transition and probably a phase inversion that's taking place here, and needs to be understood in greater detail. These results are described in a recent article in Soft Matter. And the most astonishing thing about this is that if you have plaques that aren't older than a few hours, you can reverse their porosity from this or this back to this just by shifting the pH back and forth. So I plan to do and describe to you a study that better reconciles the biochemistry that's available with the processing, with a particular focus on the phase behavior. And the resources that I have available for this, to which I owe the hard work of many trainees, because this has been created largely by us, is the proteome based on cDNA and transcriptome sequences, the protein locations in the plaque, which has been done largely by mass spec, layer by layer mass spec. And this will surprise many of you, because the typical way of localizing a protein in cell biology is by immunofluorescent methods. Well, you can't do that with a muscle for most proteins, because if you isolate a protein and inject it into an animal for antibodies, the antibodies will be made to certain epitopes that are available. And in these proteins, the reactivity of all of them is so great, and the probability that they change their epitopes is so great that they lose their detectabilities in their mature structures. And finally, because of the modifications, a lot of hands-on protein sequencing was required. We do much less of that now because it's been done, but it would have to be redone with another organism. And I'll give you some of those data as well. So the transcriptomic and the genomic data, no, not the genomic, cDNA data, muscles are not genomes, looks something like this. There are 4 major proteins that are relevant to the interface in plaques. You can see the size of the families is variable from large to only 1 member. And because I'll get back to this, I won't waste any more time on it. We have used mass spec in a layer by layer application to determine which proteins are where in the plaque. And from that, we know that MFP5 in that acronym stands for muscle foot protein, if you didn't see the definer earlier. Occasionally, there are MFP3s that are present at the interface, and they also go, the 3 variants and the 6 variants go more deeply into the matrix. You eventually get to MFP2 to 4, and you get to the collagens that extend down into the plaque. and MFP1 and proteins associated with it comprise the protective cuticle on the outer surface of the whole structure. Yes, I want to give you some specific biochemistry for 3 of these proteins because they turn out to be very important in the phase behavior that occurs in the plaque. MFP3 is a large family with over 30 variants. The masses actually range from about 5 to 7,000. This one is very close to 6,000. The PIs are extremely high and due to all the arginines present. Primarily, there's only 2 lysines here. They are intrinsically disordered proteins, and they're very modified. And this enlargement shows that in this particular variant, all of the tyrosines are modified by an ortho-hydroxylation of tyrosine. And presto, you've got a protein that contains Dopa. This family of proteins also has 3 or more tryptophanes at the carboxy terminus, and this is the residue that's largely responsible for the intrinsic fluorescence of the protein. MFP5 is the primer at the interface of the plaque, so this is the operative adhesive, though it depends on the other stuff that's piled on top of it. It also has a very high PI, but that PI is lowered somewhat by the post-translational modification of all the serines. They're never always modified. When you purify the protein, you discovered different degrees of phosphorylation. The protein is rich in glycines and lysines, only 2 arginines in this one, and replacing lysines with arginines results in lower adhesion. It is intrinsically disordered, and it is slightly larger than the MFP3 family. The last protein sequence I want to give you is MFP6. There are 5 variants of this at least. The protein is rich in glycine. It's also rich in tyrosine, but very few of these tyrosines get converted to Dopa. Usually it's limited to between 4 and 5 mol%. It's rich in lysine and arginine, so ergo the high isoelectric point. It does have an ordered region that was characterized by 2D NMR. If you would like to know that citation in biochemistry, I think 2016, I'll be happy to pass it on to you. So there is a beta sheet structure in this protein around about here. And that's why probably this 1 disulfide bond is present. There are 11 cysteines in the protein. 2 of them are engaged in a disulfide bond, and the other 9 are available as thiols. So very reactive protein. You have to be very careful when you purify that to keep it stable. So the questions I want to address in my talk after this short intro is, first of all, an easy one: do MFPs phase separate in solution? Do some MFPs undergo LLPS, liquid-liquid phase separation, in the plaque? I'm afraid my evidence for this is not as strong as it could be, but it's as strong as it needs to be for the present. Do MFP LLPS systems store chemical energy? That's getting towards the end of the talk. And finally, the model does stored energy maintain adhesion in muscles? LLPS, that is, these liquid phase separations, are well known and were considered fairly early on in the exploration of adhesion in these marine organisms. To go back as early as 2005, and maybe there's another one earlier at 2003. So that involves the production, the bioproduction and storage of two sorts of peptides, because all we knew about at that time were these complex coacervates, as they're called, that combine polyanions with polycations. And they profit underwater adhesion by making a fluid that is water insoluble. It is more viscous than water, so there are delivery challenges, but these coacervates are marvelously shear thinning. So they can be piped through some pretty narrow apertures. They also have very low interfacial energy, so they are inclined to try and surround and engulf anything that they come in contact with. They have high friction coefficients, and they deliver very surface active groups to the surface by spreading out onto it. So all of those attributes were generally appreciated, though measurements with specific MFPs, adhesive proteins, had yet to be done. Most MFPs, because of their strongly cationic nature and aromatic, I should add, undergo LLPS under appropriate conditions. And 3 of those phase transitions are shown here. One combining MFP3F, the arginine-rich precursor, with hyaluronic acid. When viewed under confocal microscopy, the droplets look a little fuzzy. However, by vertical and horizontal side views, they do spread spontaneously on surfaces. Now, there's another interesting MFP variant, MFP3S, that coacervates or phase separates on its own, a lot like Resolin does. And this produces droplets that are much more clearly demarcated and much more hesitant to spread once they find a surface. And most notably, they are so viscous that I'd worry whether they could be pumped through any orifice, although maybe they shear thin. I don't know in this particular case. The third study, done by groups in Korea and at Bayreuth in Germany, used MFP3F in combination with sulfate and found that the turbidity is inversely correlated with temperature. Increase the temperature and it clears up, decrease the temperature and it becomes turbid, reversibly so. There's a hysteresis in the process. And this really put the flea in our ear about what we should consider in the case of in situ phase changes in the plaque. All right. A few words about architecture. The plaque is intrinsically fluorescent, which is due to the high tryptophan content, but also related to aromatic cross-linking. The intrinsic fluorescence has a broad wavelength of excitation and a very broad wavelength of emission. And so FRAP bleaching recovery does not work. at least not based on intrinsic fluorescence. Possibly it can work with another fluorophore that's introduced, such as this cyproruby, that seems to like the proteins at the edges of the plaque. But what I really want to use this slide for, apart from its being pretty and colorful, is that I want to point to the edge of this plaque, which is projected here after a slight cut, that shows the wrinkled but uniform film due to MFP5 on the distal edge, and the very high degree of porosity in the structure above it. A very nice shot. And I credit Justin Bernstein for that. But let's blow this up for TEM now to ask a question. What happens if we take a plaque within 1 hour of secretion and treat it with pH3, a buffer? So this is the control. The only thing that's been done to it besides staining and fixation is it was treated with DI water to remove the salts or dilute them. So this is our control in our treated at pH 3.5. You can see that the interfacial film has dissolved, and that this electron dense stuff is becoming clear, almost as if they're enclosures, the contents of which have dissolved and gone into the buffer. To study this a little more analytically, we turn to Plaque cyclic voltammetry that uses a standard three electrode configuration, in which the working electrode is gold film with a plaque on it that the muscle put onto this gold film intentionally. So what we use for these electrodes is initially a mica sheet that's had gold deposited onto it by E-deposition, and then the gold film and its plaque is peeled away for this part of the experiment. In this experiment, we are using the Dopa that's present in these proteins as a natural redox reporter, so nothing has to be added. The cyclic voltammetry curves involve linear ramps up to a certain potential and then back. And they're shown here for two proteins, 3F and 3S. These are classic patterns, nearly reversible, but not quite. And you have to do it at low pH, of course, to get this kind of result. So we let the muscles deposit their plaques. This was a control just to show that the Dopa-containing proteins are excellent reporters. Let the muscles deposit their plaques on electrodes, and then do the CV of the interface, because it's got some solvent in it, but the proteins will be quite concentrated at the interface. And what we're after for phase change information is the time dependence of current at a particular potential for phase change. So here are the CVs. At pH 8, the interface looks fairly classic. This being the oxidation peak, this being the reduction peak. And at pH 3, the oxidation peak is much larger. And this was done on the plaque after allowing it to sit in pH 3 buffer for an hour. And it's become very irreversible, probably because of the products, the cross-linking products that the quinones go on to form. And Now we want to know actually what the time course of evolution of this current at this potential is. And here they are. This is the family of curves, 3 curves, one at without delay, one at 30 minutes, one at 50 minutes. And you can see that the current increases to an asymptote, and then things start getting messy afterwards as the system becomes more cross-linked. So this does not tell us what phase the material is at initially. It's in a hidden form. It could be a hydrogel, it could be a solid, it could be a liquid phase separated form that is less accessible to the electrode. And as the acid dissolves it, more and more of the dissolved reporter molecules are available and inform us about their presence. Okay, so as I said, this is not conclusive about what kind of phase change has taken place, except that it's gone from cryptic to exposed. But we have enough here to ask the question, what proteins are being released by the phase change? And in order to do this experiment, we simply borrowed an electroblotting apparatus from a molecular bio lab and are using a plaque instead of a membrane. We can use any buffer we like, and so we chose buffers at pH 8 and pH 3 and looked at the proteins that are transferred, because they won't transfer unless they are soluble. And so a typical result is start with a plaque. After blotting, you have a footprint. You pickle that footprint with matrix, and then you can do a high resolution MALDI analysis of that, that pickled cocktail. So here's what a spectrum would look like. And now we're getting into detailed data. So I'll be as clear as I can. The black tracing is for the acid released proteins. The red tracings are for the pH 8, so seawater pH tracings. The red would be what's soluble under seawater conditions. And note that the peaks are all quite sharp and that they can be identified, I'll tell you exactly how, by the masses that they have. We have 2 variants, notable variants from the MFP3 family, 3 beta and 3-6 it's called, and MFP6. They are present in the acid and not in the base. MFP5 is not very abundant, but then you wouldn't expect it to be. It's only there as a primer. And it's there in 2 different states of modification, the tyrosine to Dopa modification and the serine to phosphoserine modification. Now, with these spiky forms of MFP3, they're spiky because that population contains no modifications to the protein. It's just the sequence as predicted by the cDNA. In contrast, these others start from or originate from the unmodified form, but there's very little of the unmodified form in the population that changes phase with pH. Instead, it's spiky because not all of the tyrosine is converted. So let me show you that here with this table now. The first peak, that is this one, corresponds to this variant, which has this mass as deduced from the cDNA-based sequence. And the second peak has this mass from the cDNA MFP5 has this mass, 8909, and MFP6 does not, in mass spec, resolve the different variants, and they're very close anyway. So we just will have to look at that as a mixture. Now, how did I deduce this? Let me take you through that because it ends up being very important. So that peak, or at least the top of the peak, is boxed here. How do we reconcile these masses with the parent and with the degrees of modification? Here it is, just the top of the peak. Here's the sequence. The arrows point to the tyrosines that are modified. 0 modifications put you down here somewhere. And 52, 56 is the first peak in the box portion. So that puts us at 4 tyrosine hydroxylations to Dopa. And the 53, 52 puts us at 10. These are so important to what I'm going to tell you because they are not only fiducial mass markers, but there's also a centroid that's associated with this cluster, and they represent changes in redox. So that a purified protein that's stored at pH 3 for days in a non-functioning refrigerator shows a decrease. That is, the centroid still has spikes, but they drift. They drift, to lower masses. Why to lower masses? Because every time a Dopa residue is oxidized to quinone, it loses 2 Daltons in the form of 2 hydrogens. And so you can track from the mass of your protein the degree of oxidative damage that it has undergone. Okay, now here's the acid extracted peak again for that family. The black is for this family. Just to prove to you that these represent the pristine mass values for the protein, we isolated the protein from the proteins from the foot, which has never been exposed to seawater, and compared it alongside. And the third tracing, the blue one, is either black or red protein that is been cleared of any solids, but dialyzed against seawater or 30 millimolar sulfate at pH 5. Now you can see a 2-hour exposure to seawater or sulfate has resulted in the centroid drift of this cohort of proteins, but this one here remains mostly unaffected. It's a little affected, but mostly unaffected. So that tells us that being at low pH or in a liquid-liquid phase separated form protects against oxidation. That happens rather handily when the proteins are exposed directly to solvent. Now, you might ask, why did we use sulfate here? It isn't just because the previous investigators did. It's also because sulfate was detected to be very concentrated near the plaque solid interface, and the details of that are spelled out in Valois et al. So now a hypothesis that needs to be extensively tested. Does sequestration within LLPS droplets by these proteins offer stability against oxidation? And ostensibly to some proteins more than others. So to test that hypothesis, we used intentional oxidants like DPPH and periodate, both very strong oxidants, stronger than anything in the protein sequence in terms of resistance. The DPPH is particularly handy because it has a purple color, and as it reacts with reducing polyphenols, it bleaches to a pale yellow. So when it's in solution, the proteins reduce DPPH, and when those same proteins are coactivated, they are insulated from DPPH, and the purple color persists until this time, which possibly is due to the fact that DPPH has low solubility, so you have to add alcohol, and alcohol destabilizes the droplets. In the case of periodate, we're using the centroid shift assay. When there's no shift, the redox remains unchanged. So obviously, when the phase separated forms are exposed to periodate, there is no or very little oxidation that takes place. On the other hand, when you dissolve the proteins at low pH, even though they would normally be stable under those conditions, periodate oxidizes them very quickly. And when you raise the pH, you don't even need periodate because the quinone is readily formed at high pH, and the mass is increasing because of Michael-type additions to the quinones of things like tris buffer and ammonia and even hydroxide ion. All right, so here are our droplets again from the turbid form that is created by adding sulfate, and they can be dispersed or they can settle on an interface and be redispersed. But if they settle over a long time, they'll eventually form a film that's not easily dispersed. So with a half a dozen or so variants, prominent variants in the mix, how do they distribute into the droplets? Do they prefer to share or monopolize droplets? To do that experiment, we relied on the intrinsic fluorescence due to tryptophan and looked at the pattern of droplets. But this was also spiked by some MFP6 that was Alexa labeled to give it a different color. So if you focus on these 3 spots, 4 spots there in the green and 3 in the blue, but superimpose these images, you can see the spots again here and blown up here. And the data suggest that MFP6 goes into its own droplets, but sometimes it seems to coalesce with the other droplets. That might be true. Maybe coalescence is quite extensive, but possibly some MFPs maintain their own monopolized droplets. So it's gotten me to this stage. I want to plant a flea in your ear. If we have 2 different populations of Dopa, and they have different redox values, because you can stick an electrode into these 2 different structures and measure the redox potentials. And the difference leads to something like this that can be translated into a delta G, suggesting that this could be a reservoir of chemical energy for any damage that occurs in this. If you have a gating actuator. Having said that, I want to return to gap number 2, which left me in a tizzy in the sense that adhesion was only measured at low pH, okay? And it disappeared at high pH. So generally that's the way that's explained by the fact that these proteins are secreted into the distal depression in the reduced form with the hope and expectation that they will bidentate hydrogen bond to surfaces or even on the right surface chelate to an oxide of aluminum or iron or vanadium, etc. There are many metals that can be chelated with high affinity. But that expectation is spoiled by any kind of oxidative damage that occurs. And indeed, even this kind of interaction that Niels has put so much energy into will be spoiled by oxidizing the catechol. So what is our hedge against that happening? The old explanation by Yu et al. Was that when you reach this point where you've lost your adhesion, going down in pH doesn't help you one bit. However, if you add MFP6 to the interface, it can rescue adhesion almost completely in a time-dependent and concentration-dependent fashion. And this rescue is abolished by alkylating MFP6 with iodoacetate or any of the other agents that are available. But the problem with this explanation is that the redox potential of thiols indicates that they are even more vulnerable to oxidation than the Dopa is. So this is not a pathway that would be very reliable to muscles. So to build on the previous diagram that I had and include the architectural component, we have a surface, we have a priming film, we have a porous material with several length scales of porosity, and the material close to the primer has droplets, enclosed droplets in it that are full of chemical energy in the form of protons and electrons that can be contributed. So I put the MFP3s in the droplet along with MFP6 to show that because of the difference in the redox potentials, any kind of damage could be fixed by these coupled redox partners. And they look a lot like quinhydrone, except they're not stacked directly on top of each other. And the exchange would occur, and the inflicted damage would be transferred to this component within the droplet, which can become reduced in two pathways. One, by the thiols that are present, and two, by tautomerizing to this form of dihydrodopa that has a lower redox potential than dopa itself, and so could continue almost indefinitely until the supply of analyte in this droplet was completely depleted. So with that, I want to emphasize that I've really only pitched an idea, a plausible idea at you, but it comes with a lot of questions. How do LLPS droplets resist redox damage, particularly by strong oxidants? Is it their dielectric is not what we expect it to be? And when simulation and modeling experts model coacervates, they assume the dielectric of water without ever having actually measured it. Do droplets have a preference for the singlet versus triplet oxygen? Seems like a wild idea. What sort of thiol scavenging is going on in them? The catechol seems to have a role in LLPS partitioning in that those variants of MFP3 that were soluble at pH 8 had no Dopa in them, and those that phase separated had Dopa in them. Why? How? How is the redox energy gated between the droplet and the film? This is very important, and it could just be that you have to form that sort of handshaking pair of quinone and hydroquinone that is ortho to affect the process, but I have done no experiments in that direction. And finally, to make the research relevance more global, so many people are working on phase separation vis-a-vis membraneless organelles. Some of those organelles are going to involve inevitably redox. Is that redox shielded in this sort of way, or is there another mechanism? And with that, I thank you for your attention. Whoops.
Thank you very much, Herb. I think we can do a little round of applause if we unmute or. So with that, we will open the floor for questions. Again, you can either just sort of raise your hand electronically, or you can drop a little question in the chat. Or you can speak up.
Or you can go to sleep.
Okay.
Alfredo, yeah.
So I was very I'm very interested in the binding strength in the very beginning. So biotin streptavidin is like super strong in the biological world. How does one get, it's almost double, at least 50% stronger. Where does that come from? Is it actually multivalent?
Oh yes, yes. So it's a great question, and I'm sure it's part of the answer. Let's see. Yes, this one. When you have, and this in part explains why there's so much excitement about the Dopa, because the Dopa is a bidentate, orthobidentate aromatic. And by selecting that ortho relationship between the hydroxyls, they are adapting to the distance between the hydroxyls on many hydrated metal oxides, so that functionality can come in and displace the hydroxyl groups on the metal hydroxide, and you have a bidentate coordinate bond then, which is half as strong as a covalent bond, but reversible.
Well, okay, I see.
And even in the case of the bidentate hydrogen bond, you are profiting from Bell theory in the sense that a double hydrogen bond will have a lifetime that's 1 million times longer than a single hydrogen bond.
Yeah. And so just technical question, this might be a really stupid question, but this energy of absorption is so high. How are you able to keep the polymers or the proteins on one of the surfaces?
How are you able to keep it on the surface?
Wouldn't statistically they will rupture from? I suspect it's covalently bonded on the mica. I don't know how you stick them on the mica.
Oh, well, the mica, it's an aluminosilicate, right? And it has about out of every 4 surface cations, 1 in 4 is the alumina, the other 3 are silica. And it has a very, very tightly packed structure. And when you freshly cleave mica, many of the surfaces are concealed by adsorbed potassium. So you have a lot of obstacles in the way that have to be cleared off. And in fact, I'm glad you asked that question, because the paper by Alison Butler in her collaboration with me and Jacob in Science 2015 addresses how important it is to knock off the potassium before you can bind the underlying alumina.
I see. Thank you. This was awesome. Thanks a lot.
You're welcome.
Thank you, Alfredo. Are there any other questions from the audience? I'll just ask a quick question then, Herb, if people want to think of one in the meantime. The electroblotting technique you introduced to us, that's really creative, I think. So my question, I guess, well, a couple questions, but I'll stick to one. What do you think you might not be detecting with that technique? Because I imagine as you do this, it's sort of, there are certain things, like depending on how long you run the experiment and what's actually able to leave from one surface and arrive at the other, you might be selecting something. So do you have you thought about what you might not be seeing?
Yeah, I can give you a couple of answers to that. It's a great question, and it really is a very important part of the controls when you set up the experiment. First of all, when you do this, you have to run the direction of transfer in both directions to see if you're missing anything in the other direction. And it turned out that we couldn't detect anything in the other direction.
Okay.
So that just underlines the fact that in mussels, almost all the operative molecules are cations and want to move towards the cat.
Okay.
So the other points are this: if a protein that is soluble is at its isoelectric point under those conditions, it won't move.
Yeah.
So we may have missed something, but since we tried both directions, and we actually also tried smaller buffering steps than just the two that I described.
Okay.
And wasn't there another one? Oh yeah, there's one other part to that. And that would be a shortcoming in the mass spec analysis. Mass specs are not perfect. There are proteins that are not easy to desorb and ionize. And so if that was a member of the population, we might have missed it.
Okay.
And so in the paper by Valois et al., another control was to look at what proteins were present and absent by using gel electrophoresis, another technique. That would reveal anything missed. And it actually does reveal that MFP2, that isn't cross-linked yet, is soluble at pH 8 and can be detected, right? And it's a little too large to see by the multi-excitation parameters that were used for analysis.
And just one quick follow-up. In terms of molecular weight, Herb, like if say you had any, just hypothetically, any sort of small molecule components that the mussel might or might not be using, would you see that, or would they sort of move too fast in that technique?
Well, how small? Well, we wouldn't say sulfate, for example, right? And we didn't see MFP8 that moved too quickly. And probably went right through the membrane.
So it's interesting to think about what else might be there, but it's a really cool technique. It's really clever.
Yeah, it is. And it has potential. It has more potential.
Yeah, for a lot of other ways of sort of extracting from protein matrices.
Right. Yeah, thank you.
Any other questions from anyone before we give a final round? Yeah, Alfredo.
Do you think I could? So this is more of a curiosity question, but I'm trying to understand how this phase separation and the liquid-liquid, this liquid-liquid phase separation, how does it protect from oxidation? I think that's really, really interesting because it's full of water I have a few hypotheses, and one of them is actually that it sort of sequesters the oxidizing agent, and I don't know if more than it more than it shields, it's perhaps it just sticks it and doesn't let it on the surface, doesn't let it act all over. I mean, I don't know. I'm just very curious because that's a really interesting mechanism.
Yeah, yeah. And unfortunately, that was the last experiment that we did before the pandemic. So we haven't been able to follow it up, but we're very itchy too. But your intake of the information, I think, is missing the point that that the proteins that are inside the droplet are resistant to oxidation by the agent. Okay, it isn't just the surface of the droplet. The proteins that are on the inside aren't just, they may be at the surface, though I think that that would make them more vulnerable to oxidants if they line the interface with those redox sensitive groups. But one experiment that I'm setting up to address that question is to make liquid-liquid phase separated droplets just using cheap polymers, so I can generate a lot of volume, and then to add low molecular weight catechol to them. I know already from my experiments that they favorably partition into the droplets. But if they are not resistant to oxidation, then it has something to do with how the protein is protecting that redox group.
Yeah, exactly. That's what, okay, that's really cool. Yeah. I was
Yeah.
trying to, okay.
All right. Well, two short comments. I think in the interest of time, obviously people should feel free to hang up and move on. I know if Herb, if you have a few more minutes,
Yeah,
I know there
sure.
might be a few more questions for those who have time to linger around, but for those who have to leave, let's just thank Herb once again. And then Herb, I also want to thank you for once again managing to throw in a word that I didn't know, fiducial. So now I had to go look up yet another word.
Fiducial.
Fiducial. See, I can't
Yeah.
even pronounce it. So
Yeah. I
thank
thought
you.
it had to do with banking, but it's used in research with respect to control markers.
Yeah. Thank you, Herb. And for those who want to hang out afterwards, I know I have a few more questions and some other people do, but feel free to hang up if you need to go, okay? Jake, did you want to ask a question?
Yeah, hi, Herb. Thanks for the talk. So I just have a question about the oxidation of Dopa.
Yeah.
A lot of literature that I've read concerning the use in like more synthetic materials. So like, for example, they show that, a lot of times the quinones can cross things together to form didopas and polydopas, right? And in a lot of those drawings, it's drawn such that the hydroxyl groups are still there after that sort of cross-linking. And so I was wondering if that implies that they can still coordinate to these surfaces even in these oxidized states. I don't know if that's relevant to the real muscle and how it has the Dopa groups, but.
No, it's an important question because at very least because the literature is so biased into viewing the fate of Dopa as being oxidation and cross-linking. Okay, that's a bias that goes back to Lord Todd at Cambridge University in 1940. And he came to that conclusion with low molecular weight quinones that really were not very good analogs of what occurs in biology. And the other sad reality is that the cross-linking that occurs in biological systems via quinones is rarely thoroughly characterized. So I had one trainee, Ali Mizarez, who's at NYU and NTU, who did it for squidbeak. So squidbeak is, a brownish structure, very hard, sharp as a knife, and is a very highly cross-linked material. And And he isolated those crosslinks and thoroughly characterized them by mass spec and NMR. So you know that what he characterized is real. But in that system, as probably in insect cuticle, the pathway for Dopa is ultimately to get oxidized and to cross-link with certain protein partners. But with many other systems, the cross-linking pathway has not been proven. I'll accept that oxidation is very, very likely if you don't protect the system, but not cross-linking.
I see. Thank you.
Anyone else?
I had a quick question. This is
Yeah.
Lisa. I think you might have already explained this, but you showed a distribution of the different numbers of MFP proteins in the foot.
Yeah.
And then you showed a slide about the different types of post-translational modifications and the ratios of the two.
Yes.
Is there a good understanding of why you have that particular gradient across the muscle foot, and then how that relates to the amount of post-translational modification?
That is an itching question as well. At one time, I was seduced into thinking that the reason that differential hydroxylation existed was that mussels use their foot to explore a surface first, and they, based on the information that they get from that surveillance, they then adjust the hydroxylation in the proteins accordingly. But the proteins that show the highest degree of hydroxylation are not the adhesives. They are the reservoir, the antioxidant reservoir. And so now my hypothesis is that they adjust the degree of hydroxylation to tune the phase boundaries of when those droplets occur, because it's a temperature sensitive process, it's a salt sensitive process, and it's a pH sensitive process. So having that ability to tune it, and everything in biological is multifunctional, okay? That has to be said. So if they use the energy to put more hydroxyls into their polymers, they're benefiting by prolonging the lifetime of that sort of battery reservoir that they're making. But they also tune the boundaries for phase separation. So let's say there is a very heavy rainfall with lots of runoff. So the intertidal salinity will get changed by 50% from what it usually is. The mussel will then be able to adjust the phase boundaries appropriately for the phase separation. It's an idea. I think it's plausible, but it hasn't been tested.
Okay, thank you.
So Herb, following up on that, do you think it's, are you saying that the feedback, like that adjustment capacity, is that a feedback that goes through sort of the living part of the system, or do you think it's, okay.
Yeah, that would be, that's a neural networks question, right? They have a sensor. The foot is definitely doing surveying, but but the nature of the ability to survey chemistry, it makes sense for them to be able to do roughness, for example, right? And I'm sure they do, because they select a certain kind of roughness. They don't like completely smooth things. They don't like things that are too rough. They select an intermediate roughness. But to be able to sense chemistry is a little hard for me to understand, unless they have some kind of olfaction system in the foot.
Yeah, but that's also like your last hypothesis of sort of the, I don't know whether redundancy is the right word, but this sort of, or flexibility, I don't know how to articulate it well, but but it seems like just the materials component in the quasivate droplets are able to adjust and sort of have a period of time over which they might actually be able to optimize whatever reactive chemistries that need to happen to, say, for example, bind to a surface.
Yeah.
Almost as if there's I don't know, redundancy or resilience or built-in molecular chemical flexibility that allows these molecular feedbacks to also have sort of a broader window of surfaces to which they can respond to. Does that make sense? That it's
Yeah.
all the way down at the molecular level as well,
Yeah.
within the droplets, perhaps.
Yes. Well, to me, any kind of surveillance is astonishing because they live in such a high energy environment,
Yeah.
you know, where waters are frequently high velocity and turbulent, and how they find the time to do meaningful surveillance is pretty puzzling.
Yeah.
But they do.
Yeah.
And one thing that I can tell you about a discrimination or a surveying capability that they do is when a surface is badly fouled with slime, they just avoid it altogether. So that's not even a roughness issue.
So they should just coat all the ships in slime to avoid
Yeah.
fouling, I guess.
Well, that's gonna have its own slime, its own drag properties.
True. Last follow-up question then. So obviously mussels live in fresh water as well.
Yeah.
Would you dare to speculate what then might be different with sort of those water conditions, like lower salinities? And I don't know if the pH is very different, but the salinity certainly would be different, right?
Yeah. Well, you're actually tickling my mind in the sense that we know from a protein sequence that although there are homologies, the freshwater mussels use a much higher mole percent of acidic residues. Okay, and so I thought of this based on work with MFP1 in particular, that phase separation that's based on cation aromatic or cation pi interactions, that relies to a certain extent on high salinities to overcome the repulsion between the cations when you concentrate, right?
Yeah.
And in freshwater, that would make the pi-cation interactions more difficult.
Yeah.
But the coulombic ones would work.
Yeah. So there might be an adaptation to those different conditions. Yeah.
Yeah.
Cool. All right. Well, if there are no further questions, we should also, you've had a long day on Zoom already, Herb, so we should also let you go enjoy the sunshine it looks like you still have outside the window.
We do have very beautiful and clement weather here, so.
Good. You'll have to soak it up for us.
Yeah.
So thank you very much, Herb, and stay safe out there, and stay safe everybody else, and enjoy Thanksgiving break next week, everybody, okay? Safe travels to those of you who are traveling. Thanks again, Herb. We really appreciate it.
All right. Goodbye, everybody.
Bye-bye.
Bye.